Alright, here we are with one of the last topics that we'll cover in this semester. We are on to pyrimidine synthesis. Now, one of the things that makes pyrimidine synthesis maybe a little bit better or easier is that, well, if you compare the structures of purines and pyrimidines are smaller. They just have six atoms within their rings. They're just a little bit smaller to deal with. Now, the origin of those atoms, like I said, a pyrimidine only has 6 atoms within the ring, and the kind of awesome thing is that, well, nitrogen is coming from a glutamine amide, our carbon is coming from a HCO3-, and the remaining atoms are coming from aspartic acid, which, if you think about an aspartic acid, well, we've got our N-termini, our alpha carbon, then the 2 carbons that are in our R group, and those are all of the atoms that constitute the entirety of this molecule. 6 members, they're all wrapped up over the course of 3 different molecules. So the first thing that we need to talk about with respect to pyrimidine biosynthesis is we have to begin actually synthesizing our molecule, obviously, but we're actually presented with something that's a little bit familiar. We're presented with carbamoyl phosphate synthetase 2. Okay, CPS2 is a key regulatory step in pyrimidine synthesis. Now, we've actually already seen carbamoyl phosphate synthetase because that was actually our first step in the urea cycle. CPS1 was that first step that we used to put together our carbamoyl phosphate molecule from the nitrogen, or the NH3, that we got from our glutamate. Now in this case, what we're doing is we're actually making glutamate. So what we're going to do is the enzyme carbonic anhydrase arguably is going to be the first enzyme, because what that is going to do is going to combine CO2 and H2O to give us our HCO3. We're going to use 2 ATP molecules, glutamine and H2O. What we're going to release from that is glutamate. Okay, so what that means, that makes this a little bit different from CPS1, is this amino acid glutamine. Let's think about what it has. It has 2 nitrogens, one on the N-termini and one in the R group. Okay, so what we're going to do is we're going to take the nitrogen off of our R group, and that is going to be our NH3 that goes on to make our carbamoyl phosphate. So the only thing that differs between CPS2 and CPS1's reaction is CPS1 is not going to use glutamine as the nitrogen donor, but instead it's going to use NH3. But like I said, we're talking about CPS2. CPS2 is going to take the NH3 off of our glutamine R group, combine it with HCO3, and then we're also going to transfer one of our phosphate groups off of our ATP to make carbamoyl phosphate. So essentially what that means is here's our nitrogen from glutamine. This carbon is from our HCO3-, and then this phosphate is all that we have left. Okay, in addition to differences between CPS1 and CPS2, well, CPS1 catalyzes its reaction in the mitochondria. CPS2, on the other hand, is going to be done in the cytoplasm. So essentially what that means is you can look at these 2 metabolites, you can look at carbamoyl phosphate, and you could ask the question, well, is carbamoyl phosphate in the cytosol or is it in the mitochondria? Well, it's actually in both. But what is the fate of those? If carbamoyl phosphate is in the mitochondria, well, that is ultimately going to be entering into the urea cycle. If carbamoyl phosphate is in the cytosol, well, that, or in the cytoplasm, or the cytosol, that is going to be involved in pyrimidine synthesis. All right, so we've done all of this right here. We've synthesized our carbamoyl phosphate. And if you compare this flow chart or this pathway to our purine synthesis, well, it's much shorter. And that's something that everyone's happy to see because of the fact that, well, we're synthesizing a smaller molecule. It should be shorter, okay? And with that said, when we synthesized our carbonoyl phosphate molecule, we already had 2 of our atoms within our ring in place. All that we had to do was, well, essentially everything else. Now that everything else, sorry, I had a bug fly in front of my face and I had one land on my head earlier. What we're going to do is we're going to use the enzyme aspartate transcarbamylase. Now what that is going to do is that is going to displace this phosphate group. Let me go back and highlight with a different color the phosphate group. So that phosphate on our carbonoyl phosphate is going to be displaced. Okay, so that's released right here as an inorganic phosphate. The aspartate residue is going to, on its N-termini, going to be added in the place of that phosphate group. So now what we have is our N-termini, our alpha carbon, our C-termini, and then these are the atoms of our R group. Okay, so we have the building blocks of, well, actually, at this point in time, we have added by adding the aspartate residue, we've added the other atoms of our ring structure. What we have to do is close in that ring. So this carbonoyl phosphate has all the atoms that we need for that ring structure. The enzyme dihydroorotate, that's going to remove an H2O, and that's going to help us complete our ring and cyclize our structure here, because we're going to lose one of these hydrogens from our NH2, and then this OH right here. Those are both gone, in which case that's our H2O that's released right there. So dihydroorotate has our ring structure in place. What we're going to do is use quinone, and what that is going to help us do is, what that dihydroorotate dehydrogenase is going to enable us to do is going to be, in this position right here, we're going to add a double bond. That will give us our orotate. Orotate is going to bring about our PRPP, which is a common or a familiar face to what we saw in that PRPP that we have coming in there. That's phosphoribosyl pyrophosphate. Okay, so at this point, what I would want you to be thinking about is, what is that doing, and what does that structure look like? Well, what that's doing is bringing in our sugar, our ribose, plus our phosphate group there. So we're able to make our N-glycosidic linkage, and the enzyme that catalyzes that reaction is orotate phosphoribosyl transferase. So now we have the 6 atoms that constitute our pyrimidine ring. We're able to throw those onto our ribose, or our phosphorylated ribose, and in that we make orotidine 5' monophosphate, or OMP. Now that OMP, all that we need to do to kind of finish the job and get our molecule in the exact state that we want, or kind of prime it ready for use after this, is remove this darn CO2, and we use the enzyme OMP decarboxylase, which gives us a uridine monophosphate, and we're done with our pyrimidines there. Now, what we'll want to do with that is talk about and think about regulation. Okay, so regulation, what do we have that is going to be activating, and what do we have that's going to be inhibiting? Now, we've got ATP that is going to activate the carbamoyl phosphate to carbamoyl aspartate reaction. Okay. In contrast to that, so that was in E. coli pyrimidine biosynthesis. In contrast to that, we've got, well, ATP that is driving the synthesis of carbamoyl phosphate. So, I got mosquito. Okay, so that's going to be increasing the concentration of our carbamoyl phosphate. So that activation by ATP is also joined by the activation by PRPP to activate it. Now, one of the things that you could think about with respect to this is, where is pyrimidine biosynthesis taking place in an animal cell versus an E. coli cell? Well, they're both taking place in the cytosol, but does E. coli have a mitochondria? No, it doesn't. Okay, so with respect to a pool of carbamoyl phosphate, now what we've got to deal with there, and the urea cycle is not taking place in E. coli, but if you think about an animal cell, well, the carbamoyl phosphate might be something that's a little bit more stressed or limited in the cytosol because of the fact that, well, you have carbamoyl phosphate that's in the mitochondria, but you also have it in the cytosol. You're not pulling from one common pool. So ensuring the production of that with the help of ATP is probably something that is an overall necessary contributor. Now, in addition to that, is this PRPP, which PRPP is going to activate that enzyme as well. So this is something that's kind of an additional driver of, hey, if PRPP is present, well, we definitely need to get to orotate so that we can make OMP. Okay. Now, if we look at inhibitors, well, the same step that's activated by ATP in E. coli is going to be inhibited by CTP. So your final product in this triphosphate molecule, this pyrimidine triphosphate, CTP, is going to be inhibiting that step. So it's a feedback loop that's almost like only when you get to the end of all of this do we actually want to regulate it a little bit more, compared to in an animal pyrimidine biosynthesis. In animal pyrimidine biosynthesis, what you have is you have inhibitory steps or inhibitory activities by arguably all of your significant products: the uridine monophosphate, uridine diphosphate, uridine triphosphate. All of these are going to be regulating earlier steps in their metabolic process. UMP is going to be doing it for the step that immediately, or that actually generates its own production. But then UDP and UTP, they are going back and they are regulating the production of carbamoyl phosphate. So these regulatory steps are a little bit different from one another. One thing that's common of pyrimidine and purine biosynthesis, and I'm thinking specifically in animals, is PRPP is an activator. And so PRPP is going to kind of be that signal of, hey, I'm ready. I'm the phosphoribosyl pyrophosphate. All I need to do is get rid of this pyrophosphate and bring my 6 carbon, in this case, my 6 atom ring on, and I'm ready to rock and roll. Alright, well, I hope this is helpful, and I'll see you in the next video when we talk a little bit more about purines and pyrimidines and their value in different types of cancer researches. Alright, well, thank you very much. Have a good one.